everybody, I'm Tall.
And I'm Small.
And today we're going to be doing Khan Academy's MCAT Prep Biomolecules Genetic Mutations Questions section. Nailed it. Okay, first question. In sickle cell disease, a glutamate gets changed into a valine substitution. I read that arrow a little bit weird. Results in a formation of HBS molecules. So changing glutamate to valine results in the formation of HBS molecules, which aggregate abnormally and cannot adequately carry O2, have abnormally high affinity binding for O2, stabilize the wall of red blood cell against oxidative damage, or cause experience high levels of repulsion. I think Khan messed up a little bit there. Experience high levels of repulsion between neighboring HBS molecules. So I think the very first question to ask is, what is an HBS molecule? So apparently you change a glutamate to a valine and you form HBS. All right, we're talking about sickle cell. We know that that's going to have oxygen issues, oxygen carrying issues. Forms HBS molecules. I'm going to say that it's probably not going to cause high affinity binding for O2. It's probably not going to stabilize the wall of the red blood cell against oxidative damage.
Yeah, that sounds like a benefit.
Yeah, you'd expect it to be something bad. I do not even know how to read that last answer. Experience high levels of repulsion between neighboring HBS molecules. I'm going to say aggregate abnormally and cannot adequately carry O2.
I would go with that as well.
Yeah, so in sickle cell, I'm pretty sure that the whole pathology is that you can't carry O2 as well. And I know that it is a single nucleotide polymorphism.
And it is glutamate to valine because I missed that in a genetics lab quiz one time sophomore year.
So it makes sense that, yeah, it's right. They aggregate abnormally and cannot adequately carry O2. So that was a good example of us not really understanding the question even, but at least being able to see, okay, it's a mutation, it's sickle cell, what makes sense?
So for a second, I considered have abnormally high affinity binding for O2 because that would be a problem if we can't let go of it.
Yeah.
But that doesn't remind me of what sickle cell was, so we could rule that one out.
Yeah. Okay, question number 2. Which type of mutation results in the sickle cell disease phenotype? They're really going with sickle cell here. Is it conservative mutation, non-conservative missense mutation, a frameshift mutation, or codon deletion? Well, I know it's not a codon deletion. This is a good place, I guess, to say that there are a few things that you kind of might need to know going into the MCAT, like major diseases and what causes them. Luckily for biology people, we've kind of been brought up on a lot of this stuff. So sickle cell is caused by a single nucleotide, so a codon deletion is right out.
Yep.
I'm pretty sure frameshift mutation is right out because it's just a replacement, like we saw in the previous. A non-conservative missense mutation, which means that you're replacing one and you're getting a new type of amino acid, correct?
So missense is definitely that. And then nonsense, right, would be a stop codon. So
yeah.
Missense seems to be the correct thing here. I just don't remember what conservative is.
I think a conservative mutation is another way of saying... I can't remember what the other word for it is, but it's basically when it's like the redundancy in the code.
Okay.
So you replace one nucleotide for another, but it still codes for the same amino acid at the end, I think. So I'm going to go with non-conservative missense mutation.
Nice.
And we got it right. Awesome. Okay, next question.
Okay, real quick.
Oh, yeah.
A conservative mutation might result in conservation of the hemoglobin protein's function. Therefore, this is not the correct choice. Okay, so you're right. It would be something that is maybe a point mutation or something really benign and codes for the same amino acid. And so there's no loss of function or change of function.
Okay, yeah.
All right.
That makes sense. Okay, question number 3. A nonsense mutation is one that changes an amino acid from one to another, adds an additional codon to an RNA transcript, creates a premature stop codon, or deletes segments of RNA. I think you just said this one. Nonsense is stopping it, creating a stop codon. I don't know the best way to really remember that.
When I think about this, I kind of think about nucleic acids in that when I'm working with DNA, I'm creating something off of a sense strand. You know, and the complementary one is my antisense strand. So if I'm doing something with genes, I'm using something that is coding a message, and therefore it makes sense. But if I stop making a message or anything like that, I'm stopping the message, and it's nonsense.
So yeah, it's not sense, therefore it's stopped.
Yeah, there's no message there after that point.
Well, I am going to remember that now for the rest of eternity.
And it might be a good idea to kind of have in the back of your mind a couple of examples of stop codons in case there's a discrete question on it. So do you remember the stop codons?
UGA, UGG, and UUU?
So you started off really strong. So it is UAA, UAG, and UGA.
Okay.
And then UGG is not, because UGG codes for tryptophan.
How do you know that?
Meads was rough.
Okay. All right, next question. Question number 4. A silent mutation is highly unlikely to affect protein because... So a silent mutation, highly unlikely to affect the protein. The dominant allele can compensate for the silent mutation. The silent mutation substitutes the same type of amino acid. Multiple codons can code for the same amino acid, or the silent mutation does not cause a frame shift. So a couple of these are true. However, I'm pretty sure a silent mutation is what I was talking about with instead of like a conservative. So I would assume... Yeah, the weird thing is that multiple of these are true. Like, a silent mutation does not cause a frame shift. That's true. Multiple codons can code for the same amino acid. That's also true. The silent mutation substitutes the same type of amino acid. That's also true. Then the allele thing, I think we can kind of write out. But I guess they're looking for what's the most true.
So, to me, the silent mutation substitutes the same type of amino acid. Based on what I've learned, I hope that that's not true, because maybe changing a valine to an isoleucine is just, even though they're still hydrophobic, maybe one of them is just bulky enough to cause problems with folding or something. And so the redundancy of code here is almost more important. But it's good that you realize that so many of these are true, and you'd have to use your best judgment for that MCAT. What are you really asking me?
Yeah. When I read the silent mutation substitutes for the same type of amino acid, I kind of read it as the same amino acid. So it's good that you caught me on that. So yeah, I think multiple codons can code for the same amino acid. Makes the most sense. Here we go. Hey, it's right. Very nice. Okay, question number 5. A non-conservative mutation is one in which the new amino acid has different biochemical properties than the original. A frame shift has occurred, causing the production of a new amino acid. A large segment of DNA is deleted, or no effect results in the protein. Okay, non-conservative. So I'm assuming that they're talking about if you're going from something like an alanine to a glutamic acid where the chemical properties are totally different. Oh, wow, and that was the first answer that I read out. The new amino acid has different biochemical properties than the original. Okay, but then if we look at like a frame shift has occurred, I don't think it has anything to do with conservation. A large segment of DNA is deleted. Yeah, all this stuff is talking about big changes. So I would assume that it would be the new amino acid has different biochemical properties than the original.
Yeah, I would agree, because a deletion is a frame shift. So two of those are essentially saying something pretty similar.
Right. And then no effect, if it's a non-conservative mutation, even if it's a conservative mutation, you can sometimes see an effect in the protein. But a non-conservative, definitely you're going to see an effect. And it was right. Awesome. Next question. Which of the following represents a transition?
Oh, shoot. Do you remember these?
I don't remember what transition means.
It's like pyrimidine to pyrimidine or something like that, and then like purine to purine, or it's like some form of change, and I can't remember which one.
So purines are adenine and guanine.
Yes. Yeah.
And pyrimidines are thymine and cytosine.
Mm-hmm.
So a transition. Okay, so it's going to transition from one to the other, right? So well, actually, let's answer this like an MCAT question because
Okay.
maybe we can figure that out. So T to A, that would be completely different.
Yep.
That's purine to a pyrimidine. G to A, that is
Within the purines.
right. Those are both purines. A to T, that is a change from a purine to a pyrimidine. Gosh,
So
that's
I
hard.
think we can rule out the first and the third since they both change from one to the other. And I can't remember.
But here's the issue.
Oh, wait, and G and C is
Yeah.
also a change. So I think that leaves B. I think that was a good suggestion to answer it like that.
Huh. Okay, yeah, so B was the only one that had a pyrimidine, a purine going to a purine. Every other one was either a purine going to a pyrimidine or a pyrimidine going to a purine.
Yes.
So let's try it, and that's it.
Nice.
Awesome.
Good suggestion.
Yeah, treating it like the MCAT. Okay, next question. Which of the following is true of large-scale deletions? Enhanced silencing of important oncogenes due to altered methylation, more likely to result in frameshift mutations than small deletions, less likely to be fatal than nucleotide-level mutations, usually involve changes to DNA as well as to epigenetic mechanisms. So there's two here immediately that kind of got ruled out for me. One is the less likely to be fatal. That's absolutely not true. You learned that in Biology 101.
Yeah.
And then the other one was usually involve changes to DNA as well as to epigenetic mechanisms. Because if you just take out a chunk of DNA, it's not changing any of the epigenetics.
Yeah, no transcription factor stuff.
Right. It's not changing your environment. It's not changing anything like that. So we're left with either enhanced silencing of important oncogenes due to altered methylation or more likely to result in frameshift mutations than small deletions.
And I don't like that one either, necessarily, because it depends on, it just depends on the number, right? So if it's 3 at a time, then yeah, you might be less likely to. Oh no, that would still be a frameshift, so never mind.
Yeah. So
yeah.
I'm gonna say it's the more likely to result in frameshift mutation than small deletions. Especially because when you look at enhanced silencing of important oncogenes due to altered methylation.
Yeah, that makes no sense to me.
Yeah, they're talking about changing methylation, but here they're just the question is just asking if you do a large scale deletion. So
I mean, you could be deleting methylation, but oh no,
we were wrong.
It does happen sometimes.
You can so let's just check if that's it. No, that's not it. What? No way.
Usually involved. Okay, so let's read the hints.
Okay, so the answer was usually involve changes to DNA as well as to epigenetic mechanisms.
So, in addition to deleting important genetic material, large deletions are likely to affect some of the epigenetic mechanisms associated with genes, and not just the genes themselves. Okay, so they told us, they reworded the answer, and they didn't explain it really.
Okay. So they're saying that if you have a large-scale deletion, you're likely to affect some epigenetic mechanisms. Like if you just based on the size of it, you might end up clipping like a promoter region or something.
Okay, yeah, that's a good point.
I'm not liking that, but
yeah,
I guess I could see it. But why wouldn't it be more likely to result in a frame shift? Well, I guess a frame shift has to be in non-multiples of 3, right?
Yeah, that's what I was saying. It's not necessarily the size of the deletion. It just depends on the number at which we're taking them out. So.
Okay, so the answer was usually involve changes to DNA as well as to epigenetic mechanisms.
Don't go back on your gut. Sometimes you're right.
Yeah. Okay, next question. Nitric oxide is an important cardiovascular signaling molecule. It has also been implicated in DNA mutations in bacteria and in human cells, though these mutations are not always associated with cancer formation. Therefore, nitric oxide would be an example of an. Okay, so it is implicated in DNA mutations, but it's not necessarily cancer forming. So nitric oxide would be an example of endogenous carcinogen, endogenous mutagen, exogenous carcinogen, or exogenous mutagen. So if they told us that it's not necessarily involved in cancer formation, then it can't be a carcinogen.
Yeah.
Because isn't that the definition of a carcinogen?
Yep.
Okay, so it's a mutagen, and it's a cardiovascular signaling molecule. And if you guys remember, I think it's NOx, the nitric oxide synthase. We have actual proteins in our body that are specifically designed to create nitric oxide and be used as cell signaling, like in cardiovascular tissue. Which, if we're creating it within our body, that means endogenous, as opposed to exogenous. And I think if we like inhale nitric oxide, it's actually really bad.
That sounds like something that's a thing.
Okay, endogenous mutagen. Yay, we were right. Cool. All right, second to last question. What is the benefit of antioxidant compounds? Counteract the actions of damaging reactive oxygen species, catalyze production of free radicals in the cell.
Oh no.
Reverse existing mutations resulting from reactive oxygen species, or diminish direct damage of O2 on red blood cells. Okay, so antioxidant, I guess it helps to know what do oxidants do.
Mm-hmm.
So reactive oxygen species, that's a word that you hear a lot, and you hear about that causing damage to cells. There's just free electrons floating around that want to grab at stuff and cause a big mess. Point of antioxidants is to kind of just smother them, to occupy their active site, if you will, and stop them from enacting their damage. So I would say it's got to be counteract the actions of damaging reactive oxygen species.
Yes, I would agree with you. So we have superoxide dismutase or something like that. SOD does the first part, and then Catalyzed does the second part.
Yes.
To help us with that.
Yes.
So those creating free radicals would probably not be a good idea.
It sounds kind of like the exact opposite.
Yeah.
And yes, we were right.
Nice
Counteract the
job.
actions of damaging reactive oxygen species. Okay, last question. Intercalation. Ah, dang it.
Intercalation.
Yeah, there you go. I don't work in a wet lab. Intercalation of ethidium bromide into DNA results in, I can't wait for the wet lab person here to explain to me exactly why the right answer is the right answer, but
Oh no.
displacement of RNA polymerase, missense mutations, deformation of the DNA molecule, or constitutive expression of oncogenes.
So my knowledge of ethidium bromide is that it makes Small really paranoid when I'm working with it. And so I sometimes irrationally hold my breath, even though I don't think that you can ingest it in that way, and avoid it like the plague, breathing it in like the plague. But so all I remember from genetics about it, even though I use it on a semi-regular basis, is that it intercalates DNA. And so when I first read these answer choices, I was like, I don't know. They told me the part I know. I don't really know anything else after that. But the intercalation means that it kind of acts like a molecule that can get in between the strands, which is scary when we think about it. That's kind of something we don't want to do unless we want to use those genes for something at that moment. And so the simplest answer that goes with that is the deformation of the DNA molecule to me.
Yeah, that one stuck out to me as well.
Okay.
Mainly because I remember from biochem or genetics, or my brief stint in working in a wet lab, that ethidium bromide's job is to, as everyone says, intercalate, which means just kind of get in between bases.
Yeah, and mess with the hydrogen bonding in there.
So I could totally see it being deformation of the DNA molecule.
Yay!
And it's right. Awesome. All right, 90%, not bad. And we learned some stuff. Strong work, gang.